Good morning, my dear friends. Good morning. We welcome you all to the second day of this WNO NEEM Summit 2026. The technical sessions will be starting now. The technical session 4, there are 3 speakers, Dr. Rekha Warrior, Mr. Tangakumar, and Dr. Pushpavalli. And there are technical session 5 and 6. There is a small change in the order in which today's lectures will be conducted. After the technical session 4, before the tea break, we are planning to have an open panel discussion. This panel discussion was scheduled to be in the second half, but due to certain changes in the travel arrangements of people, we are advancing it to the session before the tea break. So I will request the speakers for the technical session 4 to be ready with their presentation. And for conducting the technical session 4, I request our Dr. Annadurai to chair the session, and with him will be one of the professors from Alagappa University, Dr. Balamurugan, who will be the co-chair for this session. I request Dr. Annadurai and Dr. Balamurugan to come and take the position in the dais. And the first speaker will be Dr. Rekha Warrior. I invite Dr. Rekha Warrior to come and occupy the dais. Though the time given was around 20 minutes, I request you to fix it at the earliest so that we can have the panel discussion and the questionnaire session for a longer period of time. Thank you very much.
A very good morning to all of you. Special thanks to the World NEEM organization, to Dr. Nirmala for extending this invitation to me, and also to Dr. Tarun Kant for recommending me to this seminar. So this gives me an opportunity. It's the first time I'm here. It gives me an opportunity to interact with different groups of people. I see the researchers, I see the industries, I see traders. So who have come together here to share your knowledge on NEEM and how to take forward this miracle tree. So once again, thank you for this opportunity. My talk would mainly focus on a follow-up on what was spoken yesterday by Dr. Gurudev Singh and Dr. Tarun Kant, who spoke on the improvement of NEEM, how to take forward this species, how to get the best material and so on. So my talk will be a little further, a little more advancement, and I will be focusing in my talk mainly on aza and oil, just like Dr. Tarun had mentioned yesterday about how we focus on aza and oil, because those are the commercially important aspects of NEEM. So my talk would also be on aza, mainly on aza. And here I don't need to describe aza because all of us are working towards NEEM for this aza. This is the most important commercial product of the plant. So aza, we know it is a liminoid compound. It is an insect antifeedant. It is a growth regulator used in biopesticide formulations, mainly as an alternative to synthetic pesticides. Now, the main focus of my talk is, do we actually only need the seed for aza? Can we try and see some other ways by means of which we can produce aza? Are there any problems with seed? Yes, there are some problems, though seed always happens to be the most economical route for production of aza. There are certain problems with as far as the seed is concerned. The first is the seasonal seed availability. So neem seed, as we know, it has a window wherein it is produced. It is not available throughout the year. The next is the variation in the aza content, as Dr. Tarun was showing, the variation from 200 to 20,000. So that is the type of variation that we see in Aza, especially because of the environmental and the genetic factors that are involved in it, and also because of the long maturation of the trees. When you plant a tree, it takes about 8 to 10 years. Maybe if we do some tweaking there, you do some manipulation, maybe we should be able to get the seeds early, maybe within 3 or 4 years, but still you have to wait for a longer period for the trees to flower and fruit, and then you collect them for the aza. So can we look for some alternatives which would help you to have aza continuously? So we thought we could go in for a controlled environment wherein we can see if we can control the production of aza under controlled conditions. Try and see if you can have continuous biomass generation. From that continuous biomass generation all through the year, can we get the aza? And can we do some manipulation to see if it is only aza that we need, or can we get some other compounds from this? And I was talking to some of the people here who are trading with NEEM, who say that NEEM collection, NEEM seed extraction is a very big process. There are also some hazards associated with it. So can we see if we have an alternative to the seed extraction? So before I go into my talk, I would like to dwell a little bit on the liminoids of NEEM. What are the different compounds that are present in different parts of NEEM? How are the different compounds produced, and what we are targeting on? So if you see the liminoid profile of NEEM, you can see that the same compounds are not present all throughout the plant. You have different compounds in the leaves, roots, seeds, and stems. We know seed has maximum aza. You have aza A and aza B, and also selenin content, which is high. But the other parts of the plant also have these compounds, and there are some compounds which are not distributed uniformly. Some are unique to the leaf, some are unique to the root, some are unique to the stem, and so on. Looking at the biosynthesis of NEEM, once it enters into the mevalonate pathway, squalene is produced, then it splits into two. It gives two distinct type of compounds. One is a ring intact limonide, which is very less in quantity, that is mostly present in the leaves. You also have the C-seco limonoids. Now these C-seco limonoids are produced as a result of oxidation, cleavage, or some structural arrangements which are made in the ring intact compounds. So once that is happening, then you immediately get the compounds of interest for us. So what I show in the light green, the fluorescent green, are all compounds that are of interest to us, which are the C-seco limonoids, which happen as a result of various modifications in the ring intact limonoid. So we need to see what are those compounds, or what are those enzymes, or what are those activities which are responsible for breaking the intact ring of the ring intact limonoid and converting them into the compounds of interest like the azadirachtin. So with this as the base, and as I mentioned that the profiles are not going to be the same in every part of the plant, we started our study using different explant sources. So we use the leaves, flowers, cotyledons, hypocotyls, and embryos for this study to see how we can manipulate them to get the compounds that we require. You would recollect what Dr. Singh mentioned yesterday in his presentation. Depending on what is your starting material, accordingly you will get the increased quantity. So first you modify your technology to suit your needs, and then go in for a material which has very high quantity of these compounds so that your later results are better. So look, taking these explants, we put them into a medium, allowing them to grow. So small bits of these tissues, we put them into a medium, allow them to develop into a callus. So for developing into a callus, we use different combinations of auxins and cytokinins. And we tried about 15 different combinations. Now what is a callus? A callus is an undifferentiated mass of cells. One advantage of plants, especially in plant cell culture, is plants have three distinct features within them. They have the totipotency in them, they have the ability for dedifferentiation, they have the ability for redifferentiation. So because of the totipotency, they can generate into an entire plant. By the process of dedifferentiation, from a part they become a mass of undifferentiated cells, and from this undifferentiated cells, when you give combinations of auxins and cytokinins, they redifferentiate themselves into a part which we require. Say for example, you have leaf parts, you convert them into a callus, then you can force them, you can manipulate them to behave like a seed. So that is an advantage of plant cells especially. This you do not see in animal cells. So manipulation is very easy here. So here I would be talking mainly about the flower. I take it as an example, the flowers that we have used as the explant. So when the flowers were taken, we modified the culture conditions. We were able to get different types of callus. So I mentioned there are 15 different combinations that we have tried. we had a white callus, we had green callus, we had brown callus, depending on the different mixes and matches of the auxins and cytokines. So the white callus rapidly proliferated, but when we checked for the aza content, it was less. The green callus, which again was rapidly proliferating, it was very friable also. The aza content was very high in it. And the brown callus, it started showing a browning nature with more of phenolics, and the growth was less. One point which we have to note here is a flower is a tissue in NEEM which does not contain any azadirachtin. It contains the other liminoids, but it does not contain aza. When it has been put into the callus, it has been able to show aza in its green callus. Now taking that as the cue, we took that friable callus, we put it into an initiation medium where we allowed it to grow further, and then through multiple passages we were able to increase the growth of the undifferentiated mass. Please mind you here, we did not allow it to grow into a plant. We just allowed the cells to keep multiplying so that each cell is able to produce the azadirachtin within the cells. So once we developed the subsequent mass of cell, this friable callus, we cut it into smaller pieces, and then we try to decide which would be the optimal size of a callus, that is called as a compact callus aggregate, CCA. Now this size determines whether this callus can multiply into a larger mass. So once we decided on the compact callus aggregate size, we put them into a liquid medium. Why do we go for a liquid medium? The exposure of the nutrients to the medium is larger. You have easier nutrient availability, you have easier oxygen availability, the growth of the callus is faster. And then breaking it up in the liquid medium and then taking it for a scaling process is also easier. So we adopted the route of cell suspension culture for enhancing the production of the callus in the medium. So when we try to look into the growth characteristics of this callus in a suspension culture, we realize that over a period of 5 weeks, the growth of the callus is good, meaning to say the cells are rapidly multiplying in the callus in the suspension cultures. But by the end of 5th week, slowly we see that there is a decline in the growth of the callus, meaning to say the callus is slowly starting to dry up, it is slowly starting to die. So this helps us to optimize the period up to which you can grow the callus. It also helps you to decide at what stage you have to harvest it for maximum or optimum production of the aza. So now the growth is there. We also need to see what is the amount of aza that is present during this period of growth. So for that we did the analysis of HPLC. Dr. Tarun was mentioning yesterday about standard methods of analysis. So when we did an analysis, first we did for the different explants that we had shown. I showed you the different types of callus that we got. There we observed that the flower callus was the best. Here I would like to emphasize we had taken just one tree. Because if we take material from different trees and we do the experiments, the genotypic variation would be reflected there. So we took just one tree, we took the seeds, leaves, cotyledons, every part for the explant was taken from the single tree. So from that tree we took the seeds and we analyzed the amount of aza that is there in the seed, meaning to say the natural content of azadirachtin in the tree. And what do we get in our cell suspension cultures? What we observed was the the flower callus could give as high as 50% of what was present in the seed, meaning to say if there is 100 μg of azadirachtin in the seed, we get 50 μg of azadirachtin in our cell suspension cultures. So without adding anything to the culture medium, we are able to get 50% of what is produced in the seed. But here we observed that the leaf and all other tissues that we used, the amount of azadirachtin that was produced was very minimal. Even the cotyledons did not respond well. So this was present actually in the callus. I mentioned we do different subcultures, we allow it to grow. So when we did different stages of culturing, so we transfer it from one medium to another, so we did different cycles in that. We did 4 cycles because by then we found that it was slowly dwindling down. The growth of the callus was slowly dwindling down. We realized the azadirachtin production increases by the end of the 2nd cycle, but after the 3rd and the 4th, it slowly starts to come down. So even if you're growing it for 5 weeks, maybe by the end of 2 weeks, we should be able to harvest it, or you should be able to do some manipulation so that you increase the amount of azadirachtin in it. After the 2nd week, you increase the quantum of azadirachtin and then do a harvest. So I said leaf callus, it has been very, very less. So the amount of azadirachtin that is there in the leaf is less, the growth is also very poor. So when we checked what is the amount of azadirachtin that is present in the cycles of the leaf callus from 1 to 4 weeks, we observed it is not just azadirachtin that is present there, we find the other liminoids are also growing in larger quantities. I was listening to a couple of talks yesterday where the other liminoids like you have nimbin and nimbidin, which are also of importance in the industry. So leaf callus is one way where you can manipulate to produce more of other limonites and not focus only on azadirachtin. So this method gives us a scope for manipulating all the secondary metabolites that are present in neem using different parts of the plant. So like I mentioned to you, after the second cycle, it is not going to grow up under standard conditions. So we thought we would use elicitors. So what are elicitors? They stimulate the secondary metabolite synthesis in the form of stress, in the form of acting as precursors or triggering them to produce more of metabolites. We have two different types of elicitors, biotic elicitors and abiotic elicitors. We do not use the biotic elicitors because in case you use them like the yeast extract or the fungal extract, the chances of contamination are higher. So we use the abiotic elicitors, mainly methyl jasmonate, because it is a commonly known elicitor which gives us the desired effect. Salicylic acid, we did not use it because we are not able to predict the type of action. Salicylic acid could act as a potent inducer or an inhibitor. So we used only methyl jasmonate in our process. When we used methyl jasmonate on the second cycle, where I said that the azadirachtin content was 50% as that in the seed, we found that with just 150 ppm of jasmonic acid, we were able to get 100% or more than 100% of what was present in the seed. So this is one of the best ways by means of which we can trigger the production of azadirachtin, and within 4 weeks, we should be able to get the same quantity of aza that is present in the seed within the culture conditions. So just for a testing sake, we also tried the leaf callus which was in the second cycle to see what is the amount of other limonites that are present. Here we found, in addition to nimbin and nimbidin, we are also having a lot of precursors which are responsible for the production of all the limonites in the azadirachtin pathway. Meaning to say, if we are able to manipulate the conditions within the cell suspension cultures using these elicitors, we can also manipulate them to get compounds which we desire. Like there was talk of nimbolide yesterday. So we could be able to synthesize nimbolides also in large quantities when we go in for this cell suspension culture. In addition to plain callus culture, we also went in for hairy root culture. This is a type of genetic transformation where you use the bacteria, Agrobacterium tumefaciens. This induces roots in the plants, and once these roots are produced, we try to look into what is the amount of aza that is produced. And once in the petri dishes, when you start growing it, the root is produced in large amounts. It requires a little bit of IBA into it. The root mass produced is very large. It was something like the production of penicillin from the fungus which was commercialized. So here we get large masses of the roots, and here again we find that the amount of azadirachtin is very good. Within a matter of 1 month, we are able to produce quite large amount of azadirachtin. However, one hitch which we observed here was the nutrient has to be regularly supplemented into the medium. So it is dependent upon the amount of sucrose that is present in the medium, which helps in the growth of the hairy roots. So this requires us to regularly supplement the medium with sucrose for enhancing the growth of the biomass. But when the biomass is enhanced, we also observe azadirachtin does not grow related to the biomass. Even though the biomass increases, azadirachtin is directly correlated only with the sucrose levels. So with sucrose in the medium, we should be able to have a continuous supply of azadirachtin in our medium. So when these technologies have been standardized, our next stage would be to identify different types of bioreactors which would help us to scale the azadirachtin production. So we are planning to, in the second phase which Dr. Tarun was mentioning, try to look in for what are the different bioreactors that can help us to enhance the production for a commercial scale. It is something like a fermentation technology. So we are planning to go in for 4 different types of bioreactors: a stirred tank, a bubble column, airlift and a wave. We have already done the stirred tank for medicinal compounds. We had carried out similar experiments for 5 tree species of Dashamula, where we have been able to develop callus and also the medicines directly produced from the roots. So we believe that this bioreactor technology, when applied for azadirachtin production, would be able to give us more of azadirachtin, not depending on the nature. We would be able to directly produce them. The only issues that we envisage here are the instability in the long-term culture. Say if we are planning to grow it for 1 year, how stable would the cultures be? How frequently would we have to replace with fresh amount of CCAs? The complexity of the biosynthetic pathway, as I mentioned, with different cycles, the amount of compound that is produced varies. So that we have to look into, and also how can we take it to an industrial level, say production of 100 liters or 1000 liters. Maybe we can do it for a 5 liter and a 25 liter, but when we take it to a higher level, what are the complexities that we would be facing? So that we are expecting to look into in this case. So with this, we also aim to look into the genomic and transcriptomic studies. Now, I told you we have the seed, we have the leaf, and leaf callus, and we have the flower callus. A flower is one part where there has been no azadirachtin there. So we are looking into the transcriptome of the seed, the leaf callus and the flower callus to see what are the pathways that are undergone there. In nature, it is a set of pathways that are happening for the production of azadirachtin. What happens when we grow them under controlled conditions? Where are the changes that happen in the pathways? Can we modify them for our requirement? We are also planning to look into CRISPR-Cas technology, where we go in for three different approaches. We have a knock-in, where we try to introduce AZA genes into microbes, knockout, where we block the competing pathways in NEEM, and promoter editing, wherein we try to enhance the amount of AZA that is produced within the tissue itself by targeting pathways within the seed. We are also aiming at improved bioreactor technologies and trying to look in for models which would help us to produce azadirachtin synthetically. So, to conclude, I would like to thank the National Campa New Delhi who helped us with this funding for this work, the director of our institute, Dr. Tarun Kant, our national project coordinator, and my scholar who supported me in this work. Thank you very much.
Thank you, Dr. Rekha. Now the talk is open for few questions.
So in the seed that we had, we got around 5,000 ppm of azadirachtin. In our culture, we got 6,800 ppm. I had shown it in one of the slides. When we used the elicitors, we got 6,476. So the seed was 5,000. Here we got around 6,400.
An allied question to this is, what is the enrichment you get, of azadirachtin concentration in it? See, it matters a lot from an industrial perspective. What is the composition of your AZA portion? Is it 15%, 20% or 30%, whatever it is?
Okay, here AZA is in the highest here. If you look at the HPLCs also, the other compounds are there, but AZA is maximum produced because we aimed at AZA initially. But here again, like I mentioned, in the flower callus, we get it that way.
No, what is that maximum? Is it how much percentage you are getting it? Maximum is a very
out of the different compounds, what is the
max? The limonite complex, what concentration of AZA A and B you get? That is.
I think I would be able to say 60 to 70%.
No, it's not possible.
Because we are focusing on a particular compound. Maybe this acts as an inhibitor for the other compounds.
It's very hypothetical.
No, that is why we are looking into the transcriptomes also.
Okay, let us leave that part aside. What will be the cost per gram of azadirachtin you are producing?
Okay, yeah, when you compare it with the seed extraction, it would be costly, I would say. Maybe I would be able to rate it as if I have to purify it and then I give it, maybe for the 6,000 ppm that I get, I would charge 500 to 750. Because the extraction I find is not very difficult. I just have to put it into the callus, and it is only 2 cycles that I require. So my chemicals I require are very limited there. After that, it is only crushing and taking it out using methanol water solvent. So it is not very costly for me.
If you make a 500 liter fermenter, what will be the capital cost on it? 500 liter fermenter. Okay. No, we can discuss later. So,
thank you. Sorry, I missed your presentation. The earlier part I was at the fag end, so you might have presented it. So, what was the justification of taking the flower for the callus culture, not the leaf or some other? Because leaf are known to produce azadirachtin. And secondly, you are saying from 5,000 to 6,000, but we already have accessions with 1.5%. So what is the significant lead you are getting by increasing this one? So if we target the and secondly, if we develop a method for improving the extraction efficiency. I think the whatever target you achieve through colors or through elicitation can be achieved through by improving the extraction efficiency also. So because cost versus the effectiveness, that's what I want to know.
Okay, so for the first question, I had made a presentation, maybe ma'am missed it. I had used different explants. I had used the leaf, root, stem, cotyledons, hypocotyls, flower, everything I had used. So the best results that we got was from the flower, which does not have aza at all. So we had used all the tissues, flower callus gave us the best results. And coming to the second question, your second question was on
As this efficiency of like
no, yeah,
the
about
not
using the
taking
material, you said
1.5.
about 1.5%.
1.5%.
So that also I mentioned at the beginning, that 1.5 had come from our material from Tamil Nadu, Kalangal variety. So once this is standardized, then we can decide on what material we want to use for this process. We wanted to have one genotype from which we do the entire set of experiments. So we used a tree
No,
which
the
was within...
responses varies from genotype to genotype. I'm
Yes,
sorry.
it varies from genotype to genotype. We used one which was nearest to our lab, so that we do all the experiments there. We use the seed from there, the leaf from there, the flowers from there. So if we want a
Okay,
plant,
madam,
a material...
you know, you can take it offline. We
Yeah.
are running short of time. We thank you very much for this presentation. Please, you can have a discussion later.
Later in the offline, we just want to know from the industry perspective. As of now, every industry has to test ASA AB, nimbin, selenin, diacetylnimbin and diacetylselenin, all 6 elements. So from the industry perspective, what best technology as of now, whatever have you been done in the past, can be transferred to the industry will be discussed with you offline. Thank you.
No, it's a too early stage to discuss industrial application now. It is only in the test tube stage. Maybe, you know, she has to do more work. Thank you for your
Thank
question.
you, doctor. Now I request Mr. Kumar to come on the dais to honor the stay back. Sir.
Who is the next speaker?
Our next speaker will be Mr. Thanga Kumar. Yeah, he will be talking about Ayurvasta, the science of healing textiles, weaving, health, wellness and sustainability with NEEM. This is a topic which we have been waiting to hear, because from our insecticide or fungicide area, we would like to move to the application area, and Dr. Thanga Kumar will be able to fill that gap. Welcome to the session, Dr. Thanga.
And good morning. Thank you to all. And WNO Madam Nirmala Madam invited me, and their team, they are following me very nicely. Yesterday I was here, and I had a good experience with all the speaker. I was listening all the sessions, and it gave me a abundant knowledge about NEEM. Then I like to say I'm not a NEEM scientist, but I'm very much knowingly, unknowingly using NEEM for past 20 years in our dyeing process. Okay, we are using NEEM leaves and NEEM bark. The NEEM leaves will be giving the that light ivory shade, an yellow shade will come, and we are making it into different product, and we are selling in the market. And we are using neem bark. It will have a light gray color. So I will go with the slide now. So it will be a small video where it will tell about our functioning of our dyeing facility and our units. So our brand is Ayurvastra, and we are focusing on reviving our ancient safe dyeing method, which will not pollute the environment. So our our concept is based on, first this is yarn processing, this is warping for our yoga mat production. So it's our natural warping. They are using starch. It is from Balaramapuram, Thiruvananthapuram. Daily morning they used to do, and they used to weave the mundu, which even we are using NEEM mundu also, we call as NEEM mundu. It's hand woven. So our Ayurvastra project is actually initiated by the government of Kerala, and it was presented by Professor Ravi, and who wants to develop the handloom industry. So they were adding the value added, like the herbals. We are putting the herbs. So it is like an aqua extract we put. We boil it, and we make the shade also, and we also make the fabric energetic, and it will carry the goodness of the herb. So when the user is wearing it, the skin will be the intermediate to give them a good feeling on it, and maybe it can immunize the body. So that is the concept of Ayurvastra. So this is a machine where we are processing turmeric fabric for the t-shirts. And this is a jigger machine where we can process for the, we have already standardized stunt shades using turmeric, neem, vetiver, and indigo, so many herbs, even red sandal. So these are the units. So this is the fermentation indigo process where we take from a leaf of indigo tinctarium, an indigo plant. So it is oxidized and turned into blue color. It's a very good market. So we are using the fermentation process. We are using natural nitrogen-based seeds in it. So these are the product outcome of the Ayurvedic dyed. We call this, it's a process. Instead of more than the dyeing, more than the color we make. Our focus is to just make the fabric which will be have some good property on it. Thank you. So yoga mat is our main product. We are exporting to many countries. Bedding is very skin friendly. People are saying very positive about it. So we are based in Thiruvananthapuram, and our units are in Coimbatore and Tirupur. Okay. Yeah. Okay. So we are, I told about, we are following the ancient Ayurvastra, because India, we have to be proud. Yesterday also, many people, they have taken the seed from here, and they are cultivating neem in Bahamas. I have seen, I'm congratulating them. So to my knowledge, when I was up to learning everything in English medium, everything, I was not very much aware of Indian tradition. But when I was 35, got ill. Then when I want to go for a treatment, I selected Siddha and Ayurveda. By that only I understood there is a deep in our Tamil and most probably in South India or Indian culture. So the Ayurveda, Siddha are very famous. So these people were already 1,000 years before the Mughals, before the British, they were dyeing the fabric naturally using herbs, and it is having all benefit, and they were wrapping around it. So one thing is that there is a word called Adai Moodu Sikichai, that means cloth wrapping treatment, which has been one evidence. Based on that we are going. So that is a turmeric dyed cloth can be wrapped around the wounded soldiers. So turmeric is having antimicrobial, even neem can also be added. So they are cured very fast because they are not getting the external organism. So these are the evidence which we are focusing on. So this is the concept. Ayur means Ayurveda. We are including Ayurveda. Many people will be amazed to see why we are including Ayurveda, what is Ayurveda. But we are using Ayurveda, they are using herbs for the internal and for the diagnosing and for the medicinal purpose. We are using the same kashayam we prepare and we make for the dyes. So that is why we call them ourselves as a healthy fabric. The focus of Ayurveda is to make fabric as a carrier of the goodness of health and work by interacting with skin of the users for many health benefits. Because skin, you know, it is breathable. So we actually in Indian anatomy we used to say our body is made with Panchabhuta, 5 elements, and we are working with the 5 machines, Pancha Yendram, the nose, eyes, everything, and skin is the one component which is having breathable. When the environment is hot, you will get sweat, and when you are in a cold condition, the skin will automatically warm up. So it is having a option of breathing. So for example, I can tell you even a mosquito is biting, the blood will not come out, but it will suck the blood. So there is some opening. So through that, we can work some kind of, at least we can have some immunization. That is the initial study we made. And it was 20 years we are doing, and we are getting a very positive. We don't go for any scientific study, but our Ayurveda project was clinically already tested by the government of India in collaboration with Ayurveda College. It is already in the Google, you can search it. So, and they had a study and they concluded that this kind of fabric which is herbal infused with proper treatment, it can cure skin disease and it can give a lot of health benefits. So including psoriasis like that. So they have already clinical study has happened. Okay, so based on that only we are doing in very ethical format. We are not going in a very big factory. We want to be disturbed to the village. So that concept we are working out. So Ayurveda dyeing or Ayurveda practiced in India for a long time. After foreign rulers and discovery of synthetic dye stuff, the prominence came down. So now I think maybe 5% is only they are doing natural, and some 1 or 2% they are doing like in the health everything. So all the things has gone out. Because now so that is why we are reviving it, because you know 80% of the water is already throughout the world. It is not only in India, all over the world has been polluted. You cannot drink it. So, so that is one reason we have to be very care. As you are saying, no, the NEEM can revive the word. So NEEM already into our parts, in our dyeing, in our antimicrobial finishing we already do. So the water has to be proved. That is our first concern. Neerindri Amayadu Ulaga, 5,000 years they have told in Tamil. That means without any, water, nothing can exist. Okay. So to revive the safe and dyeing is our idea. Textile industry uses water for its process. After process, the effluent is put into the natural resources. And you know, all the drainage are going into rivers. So everybody know about that. It's a very unfortunate, no management of the effluents is a problem. So that is why this concept should be promoted by you. I request to the scientific community, even after the session, I want to talk to many people who can guide us in even scientifically do the NEEM in our, how we are doing. We are doing in a traditional method, we are not having any scientific thing, so we want to have this. See, this is the Neerindri Amayadu Ulagu, because Thannir Illamal Uyir Illai. So the water source is coming. That day our kings, they have taken, without water, nothing can be lived, even the drinking. So they have formed one village, that is the unit, you know. See, there is a weaver, there is a agriculturalist, there are potters, so there will be a temple, so the water will come in and it will rejoin with the river. So this is the structure, unit structure we had 1,000 years before. So that has been scattered. Then the water will go to the next unit, and further it will go to the, you see. So there will be harmony, there will be the environmental balance will be there, everybody will have their food, the clothing, because you have the weavers, they weave, and the jewelry, they have a dance, they have architecture, they know how to build. These I'm not selling any fake idea. You can see the temples, there is about 1 lakh temples in our, so we are good in architecture, we have a lot of things. Okay, so this is the neem tree. In that, you see how the people are swimming, how they, see this is the things the environment and the people want. They need the happiness. See, the fish are floating. And in the next video, I will show you how after the 1856 discovery of synthetic dyeing, only this happened. Okay, so I'm going for the next slide. You see, the dyeing water is going inside, drainage water is going in. The first prey will be the fish. See, everybody, they think they are putting one wet agent in the dyeing, in the synthetic dyeing, not in our dyeing, because when I was initially, I used to go to the synthetic dye. They will be using a wet agent which is it is having a lot of foams, the viscosity. First they will put one set. When they are putting more on that, the fish will die. So without fish, no water can be cleaned. Whatever dead body, even in the forest it is coming, the fish will eat it and it will make into a very nice, even like a pesticide, whatever you are saying, it is a manure also. And you can drink. So, but if why this is happening, that is what my, these 20 years, even many scientists are there, many people are there, everybody know what is happening, why they are not controlling the water, the drainage can be properly controlled, why it is happening, we don't know. So I have a neem tree, neem tree is, as you know, it is having a high value, because in my young age, my father used to give me every 1 month to clean my worms, I remember that. And yesterday also one dentist from Qatar was speaking about the brushing. I used to do. After I become more, my teeth is a problem, I was using, now it is good. Okay, so and this is the evidence which people will ask, how the India, when I'm talking about India, where is the evidence? The wounded soldiers are treated using the turmeric and the neem solution. They are bonded and they have antimicrobial property, whatever the things you know better than me. So that is cured. And this is Kalari. In Kalari fight, they have a Virali Pattu in Malayalam and Tamil. That means vigor silk. The silk is dyed with kumkum. The kumkum basic is turmeric only. Using the turmeric only, they are making kumkumam. When there is a war, they have the vigority, so they wear it in the abdomen and they fight. So this is the root of all the karate, kung fu, or the Japanese, whatever the Chinese they were using. This is the martial arts called Kalari. Okay, so now the process of the fabric, I will come to the name, how we make a kashayam and made for natural. We are using all natural fiber, we never use synthetic fiber. We make mundu, all type of fabric, traditional and even modern. This is the kashayam we used to make. 24 hours it will be soaked. The goodness of the herbs will come, and we don't know what are the goodness, that is only the scientist community know, but as a traditional thing we follow it. And okay, so these are the textile process, herbal desizing, scouring, natural bleaching, and how we are making the neem kashayam, then how the processing of fabric, the wellness finishing, we are using with the neem oil, then seasoning and completing. I will show the slide if the time is short, I think. So this is the desizing, we are using soap net. Yesterday also one scientist was using soap net. So we are using Rita for the desizing. It is called, it is just to, because when they are weaving, spinning, they will be using some starch. So before making color, we have to remove the starch. That is why this will increase the surface tension. So we are soaking it in this Rita. So this is the first process. Even if you are seeing the slide, you can dye your own fabric. That is why you can note down if you want. So this is the desizing material. So this is the scouring. Scouring, people are using caustic soda in the synthetic dye, which is very hazardous and you cannot put it back in the water. We are using the just ash water. Burned ash, you boil it, it becomes a natural caustic soda. So this all is followed in our 1000 years before Indian dyeing method. Okay, so this is the ash. Then the natural bleaching is we are vellavi, we call as vellavi. We will be using limestone, we will boil it, put the fabric down, it become white. Then we will be putting in the sun, that will break the pectin and it will make more brighter. That is what we are doing for the natural white. So this is very important. The Ayurvedic herbs, which we classify into 3, already this was in a traditional thing. One is for the color, and one is for fixing the color into the fabric, and another is for the wellness. In the wellness part, you can add anything, aloe vera, neem, you can add vetiver like that. So this is the, okay, neem in, so this is, they are collecting the neem. So these are the shades which was obtained by the herb. Okay, so this is one edition which came in 2017, neem in your knickers. Okay, so I don't know about WNO also, but we were using this neem for one Japanese who is, every year he used to come, he used to bring aramiki, it's a thing. He will put it in our neem oil solution, and he will take it to Japan and he is selling it. Now he is doing his own in Japan only. So you see that it is came in the Times of India. Maybe the date is June 25, 2017. So I am related with neem. So I have to thank Madam also for that. Thank you. So, and we do aqua extracts, and everything is based on the kashayam. We don't call as a dye bath also. And this is the kashayam we make for different colors. 10 colors we used to standardized. These are the machines. This is actually we are using the NEEM oil. We emulsify it to water and we are running it, and it will be soaked for 6 hours. So we are although putting natural resin on it. We have a formula on that. It is a traditional thing only. It is not a modern. In modern, I think they are saying about the encapsulation. The core material, then binder, they are using it. They are using in padding mangling, which I also tried. I have conducted more than some 1,000 workshops. My students used to test it. They take the solution of NEEM and they will be tested, and it is all antimicrobial. So we are doing that project. This is the jigger machine also we are doing. And this is the advanced machine we are using, even 500 kg, 600 kg also we can do. All is in Tirupur. Okay. Then we are using softener. Softener is also oil based and there is no chemical in that. And this is our shading. We have to shade it and we have to put it in a dark room for 2-3 days so the color will be more. And these are the products we have already, this is launched in 2006, very successfully exported. And men's and women's wear. This is the yoga mat is our first best selling things, and our Prime Minister Modi ji had the first mat of us in Delhi. And we are very grateful for that. And even in the Man Ki Baat also he explained about our concepts. So he is a very man, he like the nature. So this is the eye pillow. For that the student came and the NEEM, we use the NEEM. So this is another associate, he is doing dyeing from us. Okay. And this is a student we are promoting actually. Anybody who are interested, they can come to our 4 acres we land. And I want to know you, there are about 60-70 neem trees. I never cut trees, and it is there. After coming here, I want to safeguard it more, collect the seeds also. Thank you. Yeah. So this is our campus, 4-acre campus. We don't cut any trees and neems are there. Thank you. Okay. Thank you very much.
We thank the speaker for this presentation. Before throwing it open for a couple of questions, I would like to know how many washes your dye can withstand if you put in a machine?
Sir, everybody asked the first question, you are rightly answering it. See, actually, sir, my experience, first we are not harshing anything. We are not doing any HPHT. We are not harshing the yarn fabric like that, even the temperature. So, the color may fade because it is sensitive to, our natural dyes are already sensitive to water, and there are some washing condition is there. We dye naturally. If you are putting in a aerial or a high bleach agent, it is not. It's a lifestyle we used to call. So they have to take care of the fabric. It will long for at least 20 washes, they are saying.
No, but when you wash it and then dried in air, the antimicrobial activity will be gone, in the first wash itself.
Yes, sir. To my knowledge, I can actually, sir. I also initially didn't believe the feeling of. See, we are natural body actually. And we are very related with all the universe, all these are there. So I am very much experiencing it. So when you are sleeping on a synthetic diet which is already dead, the cotton is dead, and you are processing for so many things using chemical, the total property of this cotton is gone. So in that way, it is more better than synthetic dye. I was seeing many, even these Ramaraj brand also, they try to encapsulate the aromatic. But it is a failure only, because the rubbing only it will come. As you said, you are right, after some time it will go. So we are not focusing on that. We want to save the environment and we want to make sure that the user should be happy. See, many people are saying they are wearing it, now they are not getting fever. I don't know how it is related to the skin, everything like that. Sir.
Activity you have been
I am not scientist, sir. Don't ask
even then you are brilliant. You are not a scientist. Do you have any of your business colleagues towards NCR side, Delhi side, who have been doing this activity?
Sir, actually we are working with Forest College. We are working with many organizations. Even on Mr. Dr. Kumaran, who I know Pushwali Madam also, he attended our workshops. So we actually now, sir, the main, if I am in the I'm not saying in the politically, if I'm in the, Modi ji is doing very nicely. The first thing we have to do is all the people has to, from the mountain has to come down. It will not happen, I know, because that is the source of our water. That is the main source of water. So even the academic, scientists, politicians, bureaucrats, they have to be educated first where we are now. 80% already lost. We are no trees, deforestation, no animal, even no lion, nothing is there. So in future, what will happen? So this is the war footing we have to do. Why they are talking about so many developments. You are not balancing the forest, you are not balancing the environment, what to do? And people are dying in 40 years with cancer. So this is the main thing. So I very much, what to say, the scientific community or the bureaucrats or the politicians should first focus on nature. Because we already have a treasure. We already have a treasure.
I compliment you for the excellent presentation, but the real taste of pudding is in eating. We know many of the properties, but it is better that you conduct some scientific studies.
Yes, sir.
And if you go to the public with the data, the acceptance will be much more.
Yes, sir, we are
In
planning.
fact, it is something very unique. I was not aware. In fact, when I was going through the list of the papers, I told Nirmala ji, "Arey, there is a typological error. Ayurveda ko aapne Ayurvastra likh diya." Then she said, "No, it is Ayurvastra only." So, pardon my ignorance about that, but that is something very great. But best thing would be acceptance India, outside India, if you have the scientific data to prove that there is no skin irritation, there is beneficial effects, and how long they last and things like that. That is only my suggestion.
Yeah, thank you, sir. That is, we are going for that, sir. Actually, it's a divine interaction because I never thought that I will come here because I was doing all my work, but Nirmala madam called me and Justin, their team, they called me. I never expected, but yesterday I got a very good information. As sir is saying, we have to go scientifically. I agreed. Please, sir.
Sir, first of all, I'd like to thank you because I am a total follower of Ayurveda. So I was very much, taken back and I was totally lost.
Thank you, sir.
My suggestion, which is actually, I don't know which you have tried, but that can increase the market is, have you tested the fabric as sir asked perfectly? How many washes it lasts?
Yes, sir. We have tested, sir. I will tell you.
No. Have you tested with soap nut water?
Yes, sir. I will tell you, sir. You
Yeah,
asked the
because
question,
that
I
is,
will tell you, sir.
Yeah, because you mentioned aerial and etc. But if you use soap nut water for washing, because already soap nut is exported outside from
our
India.
washing car is soap net
Exactly.
only. Soap net only, bleach-free soap net. We recommend to use soap net only.
So
All the
that
foreign-
recommendation has to go with your fabric. So that
Yes,
will
sir.
help.
That's
That
right.
is,
Wash
yeah,
car-
how
wash
to-
car we do, sir. Wash
That
car we
is the way then you can increase the market. That is my suggestion.
Yes, sir. Yes.
Yeah,
Thank you,
okay.
sir.
So probably you can continue offline.
Please. Madam, actually, it's like a transformation of actually, it was a government project. After the government, it was A.K. Antony was the minister, planning commission they funded, Ayurveda college they did all the clinical trial, what sir was telling right there. Then after that, who will take it to the market? That is the thing. So my partner, who is from Trivandrum, he started investing 1 crore on that. The initial product is we made the bed sheets. So it was very successful. Initially, no one know that. What is the benefit? Everything, if you say Ayurveda, they take it very lightly. Even I also, I studied in convent, I was working in IT. So I also take the same. But when I was ill, when I went to Ayurveda, then only they told how the sleep is important, how the Panchakarma is important, how we have to do morning, sun has to, because I never see the sun for so many years. So I got ill like that. Okay, so this is the situation. What madam was saying is, so the experience which I got through my body, mind, then I got an opportunity. It's a divine interaction only. So now finally,
okay, Mr. Thangakumar.
I did a lot of research on there.
Thank you
Can
for
I,
your
can I
presentation.
just make a comment, please?
It's a very, very enlightening lecture, which we have been looking forward for a long time. Completely different from what we have been. One more suggestion or
Yeah,
question?
thank you very much for your excellent presentation. And basically what you're doing is a traditional fabric manufacturing process in India. In top of that, you've added some scientific or equivalent Ayurvedic systems. Just as a parallel, I was once traveling in Africa and I came across a scenario where for malaria treatment, they were using insecticidal treated mosquito nets.
Yeah.
So all that they were doing is they were taking your normal mosquito nets, infusing it in a washing machine
There is one
with some delta methrin or whatever it is, and then it would last as an anti-malarial thing or a anti-mosquito thing for maybe 4 weeks, 5 weeks or 6 weeks.
Sir.
And then they would repeat the process. So just to pick up from there, this is something very similar but with our own Ayurveda. And if it can be done, it's a good thing.
No,
so I
even
just wanted to
like
compliment you.
your suggestion, sir, like your mosquito net impregnated with the
there is a herb called
Yes.
mosquito repellent
Yes,
chemicals.
yes.
It is being exported from India in a long way, in a large way, and I think WHO has also approved certain units in locations near Karur who have been approved by WHO for the same purpose.
So it was a WHO program only in Africa, which
Yeah,
I
this
happened
is
to be
done
lucky
there.
to see.
Yeah, very great, very enlightening, really enlightening lecture, Mr. Come, sir.
Yeah, thank you. Let us give a round of applause
Thank
to
you,
the
madam.
speaker.
Thank you, madam. Thank you, madam, for you.
That was a wonderful lecture by Mr. Tarun Kumar. We are thankful to Dr. Tarun Kumar for enlightening us about Ayurvastra. Now I request Dr. Pushpavalli to give us an overview about the neem plantation in India. Over to Dr. Pushpavalli.
Good morning to everyone. First, before starting my session, I would like to thank my organization Coromandel and WNO for this opportunity. Just I am going to present about the plantation which we have in Coromandel. Normally, as everyone knows that the neem tree is a roadside tree, or some farmers they will have in the buns or at home, like that. So nobody tried about this, any horticulture crop or agriculture crop, neem can be cultivated. So Coromandel would like to experiment this. So we started this project. We are having collaborative project with Forest College and Research Institute, Dr. Kumaran. He is helping us for developing initial development, and parallelly we are doing some research on neem. So our main objective is to supply raw material for azadirachtin production with traceability, and to establish field gene banks for neem with high azadirachtin content, and to develop package of practices for neem contract farming. So we have 1,40,000 neem trees in 3 locations in Tamil Nadu, and these trees are planted from 2019 to 2022, all 3 locations. And these all trees it is planted, these areas are having different soil types. We have red sandy loam soil, red lateritic soil, and one is just adjacent to the sea. It's like a sand, sea sand. So this, all the neem trees we are irrigating with drip. And coming to traceability, each tree we have all 1,40,000 trees. It is geocoded. We have the GPS coordinates, and for our traceability, each tree have a code. And these seeds, like yesterday, they shared that how the seeds, the trees are selected for seeds. We selected a high hazard seeds, and it is multiplied by the seedlings was raised by Forest College and Research Institute, and we planted. And all our, we can say our farms are organically certified for neem seed, fruits and seeds. And like other crops, we also do some, taking some observation on growth. So far what we observed is, our growth is, neem tree growth is, it is very good when compared to when we see the tree which is planted in the roadside or at home or farmer's field. And we do neem research also. So we are mainly focusing on spacing, different types of spacing. If you see the literatures, there are different types of spacing. Is there 4 meter by 4 meter, 5 meter by 5 meter, even 8 meter by 8 meter, 6 meter by 6 meter. So we have a lot of spacing trials in our farm, and we are doing package of practices optimization. What I can say is, from the planting until the harvest, what are the agriculture practices need to be done for this neem. And we do yield improvement trials as well as forecast. What is the estimate from neem yield also we are doing, and we are doing some harvesting productivity management. So we are in the process of developing machineries for harvest and processing the neem fruit. And we do some ecological impact. It is because of neem tree, whether there is any birds visit, everything is increased or not. We have done some project with ornithology department. And all the these trials whichever I mentioned, it is all in progress. So one of the trials which we have completed is intercrop trials. So since neem trees are planted in wider rows, we want to plant it to some other crops in between the rows. So we tried black gram, green gram, cowpea and groundnut sesamum. And we want to try with some medicinal plants also, Adadoda, Gymnema and Moringa. And we tried turmeric. These are our overview of various intercrops. These trials are conducted in 2 seasons. We have done turmeric, and this is groundnut, sesamum, cowpea, black gram and green gram. Just I will show some of the results of all these trials. Coming to turmeric, what we found is because of neem tree, because of this shade effect, all we know this turmeric requires shade. So because of this neem shade, there is an increase on the turmeric yield. Actually, we tried 2 varieties. One is local variety, another one is ICR Pratibha variety. And one more observation is shared by Synthite. It's the curcumin content in the turmeric is increased because of neem shade. This feedback was given by Synthite. So we actually sold out whatever we produced to the Synthite. Only thing, if you go for turmeric, the cost of cultivation is more. So that factor we have to take consider. If the farmer, if he wants to do turmeric, if he is willing to spend money, they can go for turmeric up to 2 years. Because after 2 years, because of neem trees roots intervention, rhizome development get affected, and further there is a reduction in yield which we observed. And coming to black gram, green gram, groundnut, all other crops, even these crops, if we raised in as intercrop in any crop, what the matter, the sugarcane or any coconut, anything, there will be the yield reduction. Same trend followed in neem tree. But irrespective of any intercrops, there is a positive impact on the neem tree growth. We took observation on neem tree height and collar dia. It is since it's the initial stage, we took some collar dia observation, and ultimately there is a improvement on the soil nutrient because of this pulses and other crops. And there are a lot of opportunities apart from neem fruit and seed. And yesterday we all discussed. One of the major thing is neem one of the tree which can absorb high carbon dioxide. So since in India it is still in it is a government policy, carbon trading is not yet in place. Once it is done, we can trade the carbon because neem has the capacity to accumulate more of carbon dioxide. So in addition to neem seed or neem fruit, selling of neem leaves, neem bark or other things, we can also sell carbon credit also to the world. And neem timber wood nowadays it is picking up very high because of this even after COVID. So neem wood have the high value, and through intercrop also we can get the additional income. And coming to contract farming, why we have done is all this for 5 years we want to do with contract farming with the farmers who are willing to come for neem planting without affecting their agriculture field. So in contract farming, what we are doing is we are supplying high yield quality neem seedlings to the farmers through Forest College and Research Institute, TNAU, Metropolium, or in our own nursery farm. And we give technical guidance throughout the cultivation to the farmers, and we give market assurance for fruits and seeds from contract farming. Before finishing my session, I want to have one video just to give the glimpses of our plantation. This is in 2019 when we at the time of planting. Now at present you have to see the neem tree in our farm. This is near the sea. I told you, no? Initially it is a sandy soil. You can visually see the sea just adjacent to the farm. Thank you. Thank you for patient listening.
Thank you, madam. Before leaving it to the audience, I have one question. What is the ideal yield of fruits you are getting now after 10 years?
No, it is not at 10 years completed. As per the literature, if you see after 5 years we will get the yield. Last year we get from one tree we got 15 kg of fruit. Normally it is what literature says is after 8 years there will be the stabilization of yield. We can get 2 to 5 kg of neem seed per tree. But we have to yet to see that what is the yield level.
And what is the aza content in it?
Whatever the seedlings we have, it is more than 0.75 aza only we shortlisted.
In your harvested seeds.
Our seeds, whatever we have 34 parents. Out of 34 parents, more than 50 parents we got more than 0.75 aza. 0.75 aza.
Okay, good. So now we can take one or two questions from industry giants.
What is the optimum spacing you found?
Actually, what we have done is maximum 4 meter by 4 meter, and we do pruning. And but optimum ideal spacing, what I feel is 6 meter by 6 meter is okay. But initial period we have to do lot of weeding operation, weed management is required if the farmers is not going for intercropping.
Okay.
You have to spend additional amount on inter-row weeding.
For multiplication, you are using tissue culture technique or seeds?
No, only seedlings.
Okay, thank you.
We tried some clone multiplication. What we found is that growth is not as much as neem seedling.
Madam, in this,
Yes, yes.
madam, while planting this, do you have any trees where you have not given drip irrigation? Because I wanted to know what is the difference in the root and the yield if it is in natural form without drip irrigation, where continuous water comfort
we
level
do
is
have,
given to the neem trees.
because of larger area, we have gone for drip. If we go for a farmer, the farmer can't afford.
I
Yeah,
don't
that's
want
why.
to go. I
Yeah,
don't
yeah.
want to go to the reasons why you are doing is. Do you have
No.
any trees in
Yeah,
the
yeah.
same area?
We have same location. That's why we have the trial without a drip also, with natural condition. We have some trees.
So
to
it will
yeah.
be interesting to know what is the outcome in the yield
What-
of oil
yeah.
and azadirachtin without comfort level of water.
Yes.
Because as normally seen in wild conditions, lot of trees give good yield. So I'll be glad if that is,
It
taken
is
as a
there.
part of study.
It is there, but that trees are yet to yield the.
Okay. Thank you.
Yes, it impacts.
So we thank the speaker for a wonderful presentation on neem farming, and it is a model farm. I visited one of the farm, so the trees are yet to yield the full, tone of seeds yet. Thank you very much, madam.
Thank you, Dr. Pushpavalli. It was a very interesting presentation by you. I request you to come and to the dais.
Dr. Nutan Kaushik.
Thank you, the speakers. Now, Pushpa, your specs, glass. No, let him come. Don't worry. As we mentioned in the morning, we are having a slight change in the order. We will be having our panel discussion now. Then we will be having our tea break. See, since we are breaking it for the panel discussion, I would like to honor the chair and co-chair. The chair was already honored yesterday with the memento. Since he cannot carry so many mementos, I would request the Mr. Ravindranathan to come on the dais to honor the co-chair who has been kind enough to be present during the whole presentation. handheld mouse, anything is there, or only the cursor? Only remote. This is next to this. Yeah, okay. On banega. On. Okay, good. Okay. Good morning, ladies and gentlemen. We are having a slight change, as I mentioned. No, this is okay. I'll use this. As we mentioned, there is a change in our order of presentation. We are having a very interesting panel discussion now. Yesterday and part of today, we discussed about the NEEM industry. Yesterday, there were a lot of discussion about the various aspects of NEEM industry, where we are and how to take it further. During the last WNO summit in Delhi, the WNO had commissioned a job of understanding the NEEM industry. Dr. Venkatesh Devanur and myself, we were asked to prepare a white paper and submit to WNO. This was a very interesting discussion which we had. We were able to get connected to some of our stalwarts in the industry. When we were preparing this, we had a lot of interactions with Mr. Pradeep Jaipuria from PJ Margo, Mr. John Matthew from Tea Stains, Mr. Ravindranathan from SS Bicam, Mr. Vikayat from Fortune, and also Mr. Gopal Sami from KGS Corporation. They were able to provide us with a lot of insights with the industry and into the various activities going on there. With their inputs, we compiled this whole white paper and presented to WNO. Of course, this is not the end of the white paper. It is only, I would say, the beginning of the white paper. It can be treated as a version 1, because based on yesterday's discussion and today's discussion and what we are planning, we can have a version 2, version 3, version 4 also. And the interesting part is, some of these recommendations or some of the plans which we are having, the organizers and also WNO would like to apprise the various government agencies about this for the benefit of the NEEM industry. So what we are today is that we are operating in the agriculture industry, where we are operating with the biopesticide, neem coated urea, and the benefits are the non-toxic pest control and nitrogen efficiency. Somewhere we are dabbling with healthcare, personal care, and industry, and also with environmental activities, which we have not done much in these areas. When it comes to the activities, the main thing is the raw material availability, which is a very big constraint, and which is a very big, the impediment for our growth. What we are seeing, what we have seen with the presentations from various speakers, and what we have seen with the discussion with the industry stalwarts, is that a lot of infrastructure development has taken place for the procurement of these in Karnataka, Tamil Nadu, Maharashtra, and Telangana. But at the same time, the massive resource of neem available in central India and also in places like Gujarat have not been fully exploited, and we have not utilized it to the fullest potential. And in the same way, we have not exploited the availability of neem seeds in other countries. Maybe the African country is one source. People from various companies, earlier they visited some of the African countries to source neem seeds, and there has not been much headway in that. And if you see the feedstock availability, like what Dr. Venkatesh Devanur mentioned yesterday, the KVIC says 18 million neem trees, but there have been a huge reduction in the number due to infrastructure development and also to various reasons. And we are facing a problem with adulteration, and also with various other impurities added to that. And where we are actually doing in our current major area of operation, approximately 20,000 kgs of azadirachtin technical gets produced. This is the pre-COVID numbers. And during the COVID period, there was a reduction, and there are a lot of infrastructural development has taken place, and the FG price has gone up because of non-availability of seats also. And what we are actually dealing is with a 20,000, even if you presume as 20,000 kg, our azadirachtin volume and the value which we are dealing with is minuscule when compared to the chemical pesticide value. We are at 0.1 billion, and the global pesticide market is roughly around 70 billion. So that's a very big gap, and that's a very big opportunity also. And even like the speakers yesterday were also mentioning, the urea mandates, urea production in India mandates coating with neem oil. So the neem oil requirement goes up to 22 million kg. Are we having that much? Even the stalwarts in the neem coating area, they were mentioning there is a 60 to 80% non-available neem oil. So they are importing, I do not know whether I should say that statement, synthetic oil, synthetic neem oil, that's what they were mentioning yesterday. So that's a very big gap, very big opportunity for us. Other challenges which we see is the global standards in the insecticide industry. Various countries follow various standards. Various countries follow various toxicological requirements. There is some sort of an imbalance in that. There are a lot of international regulations are varying from country to country. And there is a supply chain inefficiency which we have seen over the years, and it came to the fore yesterday also. These are all some of the problems or some of the scenarios which we have to live with. But how to take it forward, how to move ahead, and what do we do with that? WNO is very keen to take it forward and promote neem industry in India and take it to the global market. So to do that, what are our opportunities? Where are we? There's a huge opportunity for biopesticide, which is untapped so far, or rather we are on the periphery. We have not even touched the middle level in that. There is a requirement for neem coated urea, which is also very high. There's a huge gap existing in that. Need for neem cake and organic fertilizer is very high. So how do we address this? If you promote neem alone, we will be having a shortage. So we should explore possibilities of having a combination of neem with other pesticides and other possible biopesticides and make it into a globally acceptable product, so that along with the pesticide, along with the biopesticide, neem growth will also be there. One more area which we can tap is on the nano azadirachtin, which yesterday one of the speakers was talking about. EU MRL is one issue which we have to address, because EU has fixed a baseline MRL for azadirachtin, which is a very big impediment for exporters, especially the tea estate people. They are finding it very difficult to export tea because EU MRL restricts the azadirachtin presence. We have got huge untapped potential in pharmaceuticals, in nutraceuticals and cosmetics. many of us in the NEEM industry, we still consider azadirachtin and NEEM as an ingredient. We supply the ingredient to somebody who formulates it and makes the value addition. Many of us, we do not know where all it enters, where all it exists, where all he uses it. So from a perspective of supplying an ingredient, we should go for a value addition. Like one of the speakers yesterday was mentioning about using nimbolide for cancer treatment. So we should start working on isolation of active ingredients and marketing those active ingredients for the growth of this industry. So to understand it more and to have a very wide discussion, I request stalwarts in this industry, Mr. P.J. Margos, Direct MD, Mr. Jaipuria, Dr. Annadurai, Mr. Ravindranathan, the three of the big stalwarts, I request them to come to the dais and discuss with the audience and discuss with the people, and maybe they can help us throw more light on how to take it forward. Say for instance, if I take about, come sir, I request you to. Mr. Jaipuria is a trailblazer in the NEEM industry. When many people didn't know about NEEM industry, he had the courage to discuss with company like W.R. Grace, thermal triology, and he had an agreement with them to supply azadirachtin from India 20 years back. That was a very big leap which Mr. Jaipuria had taken. And even now he is with the Certis, which is a very big group in the azadirachtin industry. Same way, Dr. Annadurai, he has been a stalwart in the azadirachtin in the scientific side as well as in the industry side. Having completed his PhD and postdoc from Max Planck Institute on azadirachtin based for the control of various insect pests, he has moved to industry and currently is with the company called Fortune Biotech, which has the distinction of having the first US EPA approval for azadirachtin. Ravindranathan sir has been a trailblazer in his own area where he has had an opportunity to collaborate with the very big companies like Tomco and also with Agridyne of USA. So I request our panelists to enlighten the audience to make this industry more robust and to take it to the next level of orbit, or rather I would say orbital shift for the NEEM industry. Now I request, the panel is open for discussion. Whoever is interested, they can discuss with the panelists and we can be able to support them. Mic, please.
Thanks, Dr. Balaji, for giving an overview of the concerns of the industry and in the future needs of the industry. And now the people who are interested in asking specific questions, we will be able to address as far as possible what we know up to the knowledge available with us.
Yeah.
But it's a complex area and we need to address several things, and it is a growing area and things have evolved to a very great level now. I am so confident that it will evolve much more in the coming years. So now I request to all of you to throw questions to the panelists. Thank you.
So the very purpose is, though as I mentioned, we have got the V1 of status or white paper, we would like to go to V2, V3 also. So with your inputs, probably we can dwell more and take the next step in that. Over to the audience or over to the members. Yeah, okay, Mr. Gopal Swami, I didn't see him when I entered, that's why I could not call him to the dais. Thank you, Mr. Gopal Swami. He has been a veteran in the supply of NEEM ingredient, or rather the fruit and seed, to every manufacturer in India. I do not know whether he is exporting, that he has not revealed yet, but I do not know about that. And sorry, I didn't see you, that's why I could not call you. I'm sorry. You can come now, sir.
No, sir, no, sir.
Please,
Thanks.
I request you, I didn't see you.
I am farmer, sir. My question, neem tree near pulse crop, growth and seed recovery is very poor. Why reason? Neem tree near pulse crop, intercrop,
I got it.
growth is come.
See, neem has an allelopathic effect, and whatever grows around the neem tree, there will be a poor performance of any intercrop. That is what you are facing now. So when you cultivate it, yield will be less.
Thank you, sir.
Neem has its own allelopathic effect.
Okay.
So that's what actually Dr. Pushpavalli also mentioned the same thing in her presentation also. She had the same finding.
Good morning, sir.
Sir, Mr. Gopalsamy sir, I request you to come to the dais. You have been one of the contributors for the white paper. I request you to come to the dais.
No, sir. Here, okay. Two or three questions I want to ask neem plantation. Before that, we will approach Forest Department Tamil Nadu, highest Asa content in the world, old Dharmuri district. Actually, we approach Tamil Nadu Forest Department, we will cultivate more neem tree, this Dharmuri district. Why Dharmuri district? Dharmuri district is the highest Asa content in the world, 0.75 in 20 years before. Today has come down due to adulteration, due to so many reasons. First, we can step somebody.
Definitely, sir. We can see through WNO. Actually, we are approaching various agencies, and one of the major supporting organization for WNO is the Forest Department. Various the departments of forest in various states have been very good collaborators with WNO for us. We will take it up in Tamil Nadu also, and we will see how we can promote neem plantation in Dharmaburi in a big way.
Not for WNO, some companies, corporate company also they can approach in local. It is better, no, Coromandel. I think they will approach Forest Department. Already we approached Forest Department. At that time, due to age parameters, I can't do anything. That is now, Coromandel they can do some neem plantation in this Dharmuri district better. Future, it is one thing. Second thing, somebody asked question, in Gujarat, MP, this area only maximum 0.3 to 0.35 Asa content in the three states, MP, Gujarat, Rajasthan. It is 0.35 only, the maximum Asa content. In my experience, more than 40 years, I found that it is 0.35 only Asa content in the neem. Due to climate condition, very cold, very hot, that is why 0.35 only Asa content. We can't improve it is not possible, sir. Second thing, there is one parameter. EAD per Coromandel also, in 0.75, Dharmuri district, that neem seed, they took trial Dr. Gobinath and a plantation at Tirunelveli district, we found that it is 0.45 only. Due to climate, due to sand only, soil condition only, it is changing to district to district, in my view, sir.
Yeah, thank you, sir. Thank you very much, sir. We'll see how it can be.
And the second thing, sir, now last month I found that lot of information I got. It is 20% purity, technical. Nimin and Solin is very high. Nimin and Solin is very high compared to 35% purity, technical. So that is why last 1 year, very good demand for 20% purity. Due to Nimin very high, Nimin Solin also very high.
Thank you,
And
sir.
water extraction method, Nimin and saline is very less.
Thank you very much, sir. Let's see what we can add to the white paper which you are, or the status paper which you have prepared, and then take it forward, sir. Thank you very much, sir. Thank you.
Yeah.
I think it is high time we differentiate our requirements. When we talk of NEEM, everybody is talking about azadirachtin. Now, azadirachtin is definitely required in high quantities and purity and whatnot, especially for the pest control purposes. The other important constitute that we are missing is the neem oil. Suppose if the neem oil is almost same from UP, from Rajasthan, from Gujarat, from other areas, as compared to what is there, then let us identify the southern area, southern India for azadirachtin.
Yeah.
Whereas the other areas where we can get good seed and good fruit, we can have neem oil and we can have neem cake. And that itself, as we are realizing, there is a tremendous shortage of the neem oil. We can have the neem oil in the market, demand and supply will definitely change. So this is mindset that we are talking of only azadirachtin, that is something very unfortunate that I want to do.
Very valid point. Very valid point. Take it from there, Dr. Nutan.
I think I have been participating in the World NEEM conference right from its inception. And in 2002, I gave a presentation on the azadirachtin and neem oil content variability, and which was the outcome of the Novodur project, where we had done the collection of more than 2,500 neem seeds across India, not only from one state, from all over the India. And I, as a coordinator of the chemical characterization of this neem seeds, I did this because it was done in single laboratory, just to take care of the variability from lab to lab variability. Otherwise, some lab will claim high, some lab will claim low. So it was analyzed as single point. And you see there's a current science paper in 2005 where I published about the azadirachtin variability and the oil variability. Azadirachtin, we have identified that southern state plateau is best for the aza content, whereas these northern states are best for the oil contents. So you have to see your requirement. If you're looking the neem seed tree for the oil, or you're looking neem seed for the aza content. So accordingly, this recommendation has come long time back. But unfortunately, they are not, this high aza has been followed very strictly, but high oil is still to be picked up. So I think there's a need to focus on the high oil content for trees, which are highest is in Rajasthan. So as the heat is more, and as the content is affected by heat, by by water stress, moisture stress, as well as by the soil type. Yeah. So these are very well known fact, and this is published in Current Science. Anybody wants to see, this paper is available on NAT. Please go through that one and see the recommendation. And oil is very high in Rajasthan, MP and northern states. So northern states, as Dr. Vyas very rightly said, we should not reject these trees. We should use these trees for the oil extraction, and then cake for the soil amendment. Because the cake is also, like people are selling cake. The soil is being sold as a neem cake, very poor quality. So my question was, how do you address, white paper is addressing the quality issue? How do you monitor the quality? Because anything is being sold in the name of neem, neem seed, neem oil, neem cake. Any aza, any oil content, or even the soil, they are just packing as a neem cake by mixing just 1-2% of the neem cake. So how are you addressing the quality issue?
The 2 points raised by Dr. Vyas and Dr. Kaushik, very valid points. We are not only concentrating on high aza content or producing more azadirachtin content, and the tree gives as it is, and we are talking a lot about climate change, and the tree flowers in odd seasons and fruits in odd seasons, sometimes indeterminate fruit formation Last year I saw practically the tree was fruiting at very indeterminate way. Even this month we are getting some fruits, very small fruits. So this is all we attribute everything that is happening to climate change, and this is really happening, but we don't have any proof. The second point is WNO being a now nodal responsible agency for NEEM researchers, as Madam said, the quality issues, on neem cake and neem oil are very important. Yesterday I was shocked to hear there is a synthetic neem oil. And I don't know how to define that synthetic neem oil, which is being used in urea coating. And that was, announced by one of the neem largest producer of urea. So we should, really investigate what is happening in this. The actual demand of neem oil, is only supported 65%. Rest of 35% is what oil. And the third thing is about the neem cake. What is called as neem cake is not neem cake. Many people sell something which is colored like neem cake and smells like a neem cake. Original neem cake with the fertilizer control order is having very strict specifications, and how much sold in the market is really following to that specification is a very big question mark. And when you adulterate the market and get the name spoiled in the midst of farmers, we will never be able to sell neem cake in the future, in coming years. So WNO should take up in the white paper itself, a responsibility of certifying these various brands available in the market, whether Karnataka, Andhra, Tamil Nadu, anywhere. And this nodal certificate will again recertify the FCO order, and then we will guarantee the quality of the neem cake that is circulated in the market, as well as neem oil for a charge. And also WNO is not a charity, it is not Ramar Madam. We have to do something, So we will take it as a professional responsibility, certify it properly, and create an awareness and confidence level among the users. And this is my strong view.
Thank you.
And
to
I
add
think
to add to Dr. Nutan, we will do, yes. Yeah, we will write. Nutan, to add to your earlier point of sample collection, 2014, 2015, 16, as part of my earlier assignment with another company, we commissioned a survey in 10 states in India. We collected close to 5,000 samples across the country: neem seed, neem fruit, leaf, bark, and also the soil samples. And we had a GPS marking for each tree where we collected, and the shape of the leaf, shape of the fruit, shape of the twig, everything was photographed, everything was recorded. We analyzed nimbin, salanin, diacetylnimbin, diacetylsalanin also, apart from azadirachtin A and B, in that, to know which part gives what product, which part gives which ingredient. So that was done, but being a commercial organization, we could not publish it in any paper, but that paper was presented by one Dr. Vinay Panda in the previous WNO, because it was commissioned by EID Parry at that time. That was done, and I think we can take a leaf out of your survey and also the previous data and see where we can identify the, say for instance, like you correctly, like what Dr. Vyas mentioned, if you want to move away from azadirachtin A and B, if you want to think of, say, nimbolide or nimbin for pharmaceutical application, which area should we concentrate? Which area we can concentrate and where we can get that product? So that level we should graduate now. Sorry, Dr. Tarun Kant has a thing. Yes, Dr. Tarun. You can take the hand mic. Hand mic is there. And incidentally, that one of those, the high SR seedlings or high SR seeds was also clonally propagated for future propagation in that. Yeah. It's working.
Thank you. In fact, first of all, I would like to comment a little bit on this issue of levels of ESA in oil, particularly oil, as Nutan ma'am has already said. In fact, as I had presented in my presentation, which was a very detailed presentation of a pan-India project that we did, in which Dr. Nutan Kaushik ma'am was also involved in it. She did our entire 3 years, 1000 CPT data of ESA. Oil we also did at the same time, and I had shown 2 graphs, one of ESA and one of oil. And the oil was one of the most stable characteristics in NEEM for doing selections. And as I had shown in my pan-India Indian image, map of India, it was very clear that the ESA is more focused in the south, but the oil goes up to central India and western India. We have got very high oil, and even ESA also, even in Rajasthan, there are stray genotypes which are showing very high oil content. So, gentlemen, I don't remember, somebody was saying that the ESA is very poor in Rajasthan. It's not the case. It is very genotype specific, but I think the environmental edaphic conditions and many conditions influence the genotypic effects. So that is where the focus has to be. Then one more thing is there was also a talk on allelopathy. Somebody was saying that there is allelopathy. I think this is something very not very well understood yet, and blaming lack of production of an agricultural produce because of NEEM being there and calling it allelopathic effect, I think is very premature. We need to do more research, but the research so far done by IFGTB and by Dr. Singh's group and in fact his own work that has gone in agroforestry systems, he found that the reason why there was lack of growth of agricultural crops, or maybe some problem with which is being blamed as allelopathy, is not allelopathy, it is the shade effect. The neem trees have a very big canopy. They give shade and the shade regions don't receive sunlight. And that is one of the reasons why it's not the allelopathy, it is the shade. And if you do pollarding, or if you do trimming of the neem canopy, there is enough light, and that effect is nullified. So we have to be very careful when we blame NEEM for allelopathic effect. It may not be the case. We need to do more research. And let me also tell, because industry is sitting here, at AFRI Jodhpur, at AFRI and our campus, and there is a campus that we have at AFRI in Jodhpur. And we have other experimental sites and campuses. We have around 3,000 neem trees growing in our campuses. It is like an avenue tree. And whatever neem leaves are falling down, we are collecting those neem leaves. We are converting them into, we are composting the neem leaf, and we are having neem patti khad. We call it neem patti khad. It's neem leaf compost. It has been evaluated with trichoderma, and we have done we have analyzed the content, the nitrogen, phosphorus, NPK content, and we are using that in our experiments, the neem patti khad, and it's giving us extremely good results because of the bioactives in it, the neem leaf. So that is something, and we would like to further mass multiply this product with the help of industry. Probably. That is a model that we have created, and it is selling like hot cake from our nursery. But we are a government organization, we have a limited setup. So neem patti khad, how we are making it, that is something which we can discuss with industry.
Doctor. We will take it up, Doctor.
Yeah,
we'll
and one more thing, just one last thing is that I just want to, at this point, I want to bring out one thing. That if we look at the China model, because China has, from 70s to today, in these years, in 50 years or so, China has suddenly become a big global power, industrial power. And if we look at their research funding model, we'll realize that in China, it used to be government funding initially till 70s, but things changed. That 80% of the funding is coming from the industry. It is industry-driven research. And I think if we do start doing that, taking that as a very successful model, and if industry starts supporting the private, the public organizations which are full of resources, full of academicians, researchers, scientists, maybe new industry coming in for funding.
No, already like what others mentioned.
Yeah.
Companies like Coromandel is already doing that with a lot of interaction with academia. We'll definitely take it up.
I think we have to invest 550 billion dollars is funding by the Chinese industry to the research. I think that is missing here. So I think if in India we can do that, we can, I think, topple over what China is doing.
We'll do that, and I request Mr. Jaipuria to say a few words.
Yeah, actually that
Because
is
he has
for
been,
Japuria
he
sir
is-
only
he is
that.
he has been always silent and is a very silent player.
Yeah.
He has been the forerunner of this industry in India. So I want him to add few words
Thank you.
and give us a direction as to how we can proceed further.
Yeah.
You're giving me a very tough ask because as a businessman, my habit is to listen and then take an extract out of that. I'm in the extraction
No, we would
business
like to
and do something
that
good with
we
it.
want to have a role reversal now.
Okay, so I think if you will permit me, I'll just take a couple of minutes
Sure,
giving
sir.
you a brief history of what we sort of saw with NEEM, because I have been in the NEEM business from 1976 as a matter of fact. And like what everybody was mentioning, no, you have basically in those times you had two products. You had NEEM oil and you had NEEM extractions. Now for the NEEM oil, that is the time when India was getting into a situation where we were becoming oil short. To sell the oil was never a problem, because our company started processing NEEM oil and NEEM extractions 76, 77 onwards. So selling the NEEM oil was never any problem. I have gone to Mr. Vyas when he was in Godrej to get him to buy our NEEM oil, and he said no problem. Government of India was also supporting minor oil seeds. But if, as anybody who has been in the NEEM business or any oil seed business, if you take an oil seed and you only try and sell the oil, unless you get a good realization for your extractions, your economics are not going to work out. So what we noticed during our initial periods of growth was that the volume of business that we were able to do was entirely dependent on the amount of extractions we were able to sell. If I am not able to sell my extractions or my NEEM cake, I can make my NEEM oil, but I am not going to be commercially viable. And that is where good friends like Ravindranath stepped in and helped us market our NEEM cake in a big way in the south. So over a period of time, we sort of started reaching volumes, and at one stage of time, we were doing 20,000 tons of NEEM a year in our extraction plant. But then, as times change, we also realized that the oil was being sold to soap companies. Very small quantities were going to, pharmaceutical companies, but that was only cold pressed oil. They were not taking solvent extracted oil. And that's how the sort of business grew and came up to a particular level. But we realized that our extractions were being sold as an organic manure, which had quote unquote natural insecticidal properties. So we had realized by that time that there is something else in the NEEM seed which can give you much more value than what just the NEEM oil or the NEEM extraction is going to give. And that's when, as a small company, we started getting in touch with researchists all over the place, got to learn about azadirachtin, and started preparing ourselves for it. So when the opportunity came, when W.R. Grace came to India, saying that, look, they want to get a vendor who can give them commercial quantities of azadirachtin, we were interested in doing value add, they were interested in sourcing the product. It was a natural fit. And then PJ Margo was started as a special purpose vehicle to meet the needs of W.R. Grace for azadirachtin. Now, here, we were fortunate that we were in Bangalore, and when, like all of you did that survey, we also checked, because we were buying oil cake, I think oil seed at that time when we were doing 20,000 tons, all the way up to South UP, Madhya Pradesh, Rajasthan, no, because in my home state, people used to consider it, I'm not going to go and pick up the Nimoli, it's beneath my dignity. So we used to pick it up from there. We have bought seeds from Orissa, we have brought seeds from Andhra Pradesh, all over. So we had a fairly good idea from where the seeds are coming. And when we ran it through internally for checking for azadirachtin, we found that we were sitting at the right place in Bangalore. So that is where we set up our azadirachtin unit.
Yeah.
Because Tamil Nadu, parts of Tamil Nadu, Mysore area used to give very good quality of seeds at that time. And that's how the business started. So it is important to map all these things out, but when anything that you do over here, either you will have to be very, very small when you are taking a new type of a product and then grow from there. Because a lot of people get into business thinking that you're going to have scale immediately with a new product. That's not going to happen. If you are going to turn around and say, I want Nimbolite, for example, you are not going to make more than a few kilograms to begin with. And if you are going to put a commercial scale plant, it's more like a laboratory plant. A PhD is going to make it for you, and the product is going to be expensive. So unless these things are sort of slowly sorted out and you have the patience for it. Patience in this game is very important. Persistence in this game is very important. Because if you don't have that, commercially it will be tough. So this is one bit that I was wanting to say. So like say for example, I have a question which I wanted to ask yesterday, but somehow it's on the NEEM urea coating, and probably Dr. Saab is here. He'll be able to throw light. Because he was one of the first people to put, and our company also made a product. We used to call it NEEMix, we used to call it something else. NIMIN. And at that time the authorities shot us down because they said that, look, I'm giving subsidy for urea. And if you coat it with NEEM, the nitrogen content goes 0.2 or 0.01% below the threshold value, so the subsidy will go. Everybody knows in India, if the subsidy goes for urea, nobody was willing to buy it. Now that they have mandated it, it is okay. So that is how the whole thing started. So my question, Dr. Saab, for you, I'm reversing the role a bit. Yesterday I got totally confused. Now, I do not now know what is causing the nitrification inhibition effect. Is it the oil or is it the triterpenoids? Because if it is the triterpenoids, then you need the azadirachtin, you need all the other constituents in it. If it is just the neem oil, then why neem oil? You can take any lipid and then do with it. So a rough calculation that we did at our end, because suddenly we saw a lot of small azadirachtin plants mushrooming, where the quality of azadirachtin is not good, very, very low purity. And then when we started investigating, we found out most of the stuff is going to the people who are making urea coating agents. And the neem oil obviously is not available to the extent of quantities that we are talking about.
Exactly.
So some other oil is being doped with azadirachtin to bring it to that particular content and then being sold and given for new urea coating. So it's a technical question which I would very much appreciate an answer from you.
Yeah, Pradeep very rightly said. There are two aspects. One is the physical coating onto the urea. That could be done by any oil. And that prevents the hydrolysis of urea the moment it is applied in the field. That is the first purpose. The second thing, very well established for the last 5 decades, that these are the neem triterpenes. Interestingly, somebody can suit me, but I would stand to say azadirachtin is not required for nitrification inhibition.
Absolutely.
You see, but still, when you go to the government, I'm sorry, I'll take little time, you will be.
This is, Dr. Saab, this is very interesting because when you are saying 22,000 tons of neem oil, and we are talking in terms of developing the neem oil as a primary product, everybody needs to know this.
Yes.
So, we are talking about this particular thing, and the nitrification inhibition selenin, nimbin, deacetylselenin, these are the some compounds and rather the mixture of compounds which are responsible for nitrification inhibition. Now, I am sorry to say government has really made mess out of it. One of the very senior official I was sitting with, and I said, sir, you are a soil scientist, I am a soil scientist. ICR itself has recommended at one time that 20 kilogram of neem cake for 100 kg of urea will give the best results. And then finally, when we develop the product, there is no availability of so much of neem cake. 500 gram of the material, like extract, neemix and neemin for 50 kg of urea and excellent results. There was the high nitrogen use efficiency improvement by almost 25%. All India coordinated rice improvement program. Now they are talking of 600 milliliter of neem oil. That also quality not known. That is, somebody said synthetic oil, some other oil. I dope it with azadirachtin or some other compound. So I think somebody has convinced Modi ji that there is non-agricultural use of urea. And if you want to prevent that, you do the neem oil coating. And that's how it has been going on.
Thank you,
If
sir.
I may, I'll
Thank
just
you,
say
sir.
the positive side of this order was
Yeah.
a very simple one.
Yes.
A lot of this subsidized urea used to come to the market, go into industry.
Industry,
The
that
guys who were
stops.
making paints,
That-
they would buy the subsidized
that-
urea.
that's right.
The guys who were making formaldehyde, urea formaldehyde resins, they would buy it. So that market and a lot of it as fertilizer or whatever it is would go to Bangladesh, would go to Pakistan.
Even now they can use it
Yeah.
because only the bag is urea coated, not the urea.
Now, Mr. Ravindranathan,
Anyway, so
Mr. Ravindranathan, your views, please.
I have something to say about it. See, I am basically a neem oil and neem cake manufacturer for a long time. We have been buying lot of neem fruits from northern India, central India, Gujarat, Madhya Pradesh, all these places. My only request, even I have been telling this to Dr. Nirmala Kothari earlier also, see the problem what we face in the material is not azadirachtin or oil content or anything. According to me, the material we receive from Rajasthan or Gujarat had azadirachtin, also good azadirachtin in the initial stages. But when we store it for some time, it gets deteriorated, sir. See, there in Tamil Nadu there is no problem because Tamil Nadu people, some or other, Tamil Nadu, Karnataka and Andhra Pradesh, the people who collect this neem fruits, they take some precaution in saving the quality. They are concerned about the moisture or fungus formation or getting burnt or something like that. Because they take everywhere they go, we can see some people drying some neem fruits or neem seeds in the roadside.
Yeah.
That was the practice. Doctor will accept that. But in northern India, I go for purchase. Procurement, I used to go to Gujarat, Madhya Pradesh and all. There, I see heaps of neem fruit with the moisture of about 30 to 35% also heaped in a place. What happens is that after bagging the material in some HDB bag and transporting the material from Gujarat, Dawood or some other places to Salem or any parts of Tamil Nadu, it takes minimum 4 days the material to travel to this place. Within 4 days the material gets heat
Burnt.
and when it gets unloaded, it is half burnt and lot of impurities. All these causes the damage of the oil, active ingredient and also the quality of the cake. So my request is WNO can take an initiative to educate the people in northern India. I know some of the people have come here, suppliers of neem fruits from northern India are here. So they have to take a precaution. They have to see the material has to be at least moisture should be brought below 20% and to cleaning to some extent. Definitely it is a cost adding matter. They will get the
Definitely, sir.
advantage.
Sir,
Yes,
may I add here regarding this point, sorry. Regarding your point, I think it's very valid because primary processing of the seed is very important. And Dr. Gurdev Singh is sitting here and he has standardized a method for primary processing by which you can get a very good quality seed. I myself have seen that I have received the seed samples which looks very fine from the look, like with the seed coat and the kernel inside. They look very fine.
Yeah.
But when I crush them, the kernel is totally infected with the fungus. So if I use that fungal infected seed, I am not going to get anything. The oil content is poor, the azadirachtin content is poor, everything is poor, the limonoids are gone. Actually, they are eaten by the fungus already. So this quality control starts from the primary processing.
Yes.
So training on the primary processing is very, very important. Dr. Gurdev Singh has already developed a very good protocol for the
Definitely
primary
we'll
processing
do.
of the seeds,
Yes,
and that can be taken up
Very-
further and multiplied further.
Very, very
My
valid
request
point.
is, my
No, very-
request is the people who are bringing the material or collecting the material, they, of course, the farmers collect the material, bring to a trader. The trader has to educate the farmer that you see this, you have to dry the material,
Yeah.
clean the material and bring it. So if something is there, definitely the value of northern India neem seed fruit also is very high because now the demand for the neem fruits and neem seeds are very high all over the country.
This
So
will,
this material we cannot lose. So I request that kindly the people who are here from northern India, collectors people, please ensure that you train your people to dry the material and bring it and that quality will
Yeah.
be good.
Definitely, sir. From WNO, we will have this as one of the mandates to train people who are the source or the suppliers of NEEM seeds to various companies.
I-
We'll
I-
take as one of the steps we'll take, sir,
Yes, sir.
definitely.
Yes,
sir.
Because
Thank
you have
you.
been a stalwart in that.
If I can just make a small comment here, in the south we are a little blessed because if you go south of Bangalore also, you're getting into a rain shadow area. So once it's in a rain shadow area, then when you have this fruits are coming in July, August, it's much easier to dry. You go north, you're in the midst of the monsoon when the collection is taking place.
Yeah.
So if we are wanting to get that material, we all have to sort of agree to, also pay a higher price for the seed.
Yeah.
So that
Yes.
equation of balancing, what is the price the farmer is willing to pay for azadirachtin? What is the price? As far as the urea quotas are concerned, they have a certain price for oil. For them, it's a commodity. The lower, the better. But if we have to give them the oil by getting the seed and so on and so forth, the price is going to go up. So that you will have to sort of accept.
Thank you. Thank
Same
you,
thing
sir.
for the cake. So it's
Yeah.
very important as to where you are, which is going to also determine how you dry and what you do.
Yeah, there is one more.
Yes, please.
One more part. Yeah.
थैंक यू मैं जिगरेल पटेल
But
हूँ
my
देव
request is,
एंटरप्राइज में
see,
से।
other session is still waiting, so let us make it very
मैं
short.
आपको एक WNO को एक हम ट्रेडर की ओर से आपसे एक बात करना चाहते हैं कि कलेक्ट के अंदर प्रॉब्लम है और ट्रांसपोर्टेशन के अंदर जो माल आता है वो ट्रांसपोर्टेशन के अंदर हीट होने की वजह से तमिलनाडु या कोई भी स्टेट के अंदर उसका मॉइस्चर बढ़ जाता है निम्बिन ज़्यादा होने की वजह से उससे गर्माहट से वो बिगड़ जाता है तो कलेक्ट के अंदर भी प्रॉब्लम है तो हम आपसे गुजारिश करते हैं कि WNO हमको सपोर्ट करे हर एक कोई भी जगह पे तो उसको कलेक्ट कैसे करना है फार्मर्स को इकट्ठा कैसे करना है वो एक्चुअली हम सब कर सकते हैं हम सबको बुला भी सकते हैं एक जगह हम आपको सहयोग करेंगे आप भी हमको सहयोग करके जिससे
Great.
बिजनेस
It's a
गुड हो जाए
It's a win-win situation. The industry gets benefit, you get benefit.
Sir,
the whole-
one more
yeah,
thing.
we'll
Sir,
definitely
here
do that.
I would
Sir,
like
one
to
more thing.
add.
Yeah.
Yeah,
Sorry.
on behalf of the Indian Council of Forestry Research and Education, Dr. Tarun, Dr. Singh, so if WNO is ready to support this, we would be happy to associate, ICFRE would be happy to associate with WNO and give training to the farmers, the
Definitely.
villagers who are collecting the seeds. We can go there, give a hands-on. We can create a trainers group who will then go to the different places where the seed collection is happening.
that will be
We
very-
can help in that process.
Will be
Because
very
as
useful.
Dr. Nutan was mentioning, we have been doing this regularly.
Yeah.
And the quality of seeds is really
Exactly.
good when we give it. So
No.
if WNO is ready to support, our headquarters will be able
Sure,
to do
sure,
this.
because-
We have 9 institutes across the country. So wherever you want this, our institute should be able to come forward and help you with
It will
this.
be very great
We are ready to
help
support you with that.
because the scientific diligence with which the training can be imparted will help the industry a lot. We'll definitely try that. There is one more I think Nutan, there is
Sir, one
no,
more
we have
request.
done it. Yeah. He has already developed a video.
Sir, I am a trader and manufacturer also in MP, and I am doing from 2005. Since जिस समय पे कहीं कोई कलेक्शन नहीं होता था, मेरा एक रिक्वेस्ट है सर कि दोनों तरफ से काम होता है. अगर मैं अकेला बोलूँगा कि मैं कलेक्शन के लिए बहुत अच्छा माल दूंगा और इंडस्ट्री बोलेगी कि भाई हमें अच्छा माल का पैसा नहीं देना, आप लोग जो माल ले रहे हैं, फर्स्ट ऑफ थिंग कि इंडस्ट्री को सबसे पहले पैरामीटर्स तय करना चाहिए. जिस तरह से हर एक इंडस्ट्री में होता है, मान लीजिए मेज है, सोयाबीन है, हर एक चीज़ में मॉइस्चर कंटेंट्स हैं, उसकी इम्प्यूरिटीज है, उसका पैसा दिया जाता है कि 15% मॉइस्चर है, 2% इम्प्यूरिटी, उसका पैसा अलग होता है. अगर 17% मॉइस्चर है, उसका पैसा अलग है ना. नीम के अंदर इस तरह का कोई पैरामीटर कोई भी इंडस्ट्री सेट नहीं कर रही है अभी फिलहाल में. दैट इज वन थिंग फॉर नीड. अगर साउथ के माल का पैसा अगर 30 रुपए है और एमपी के माल का पैसा 15 रुपए है और ट्रेडर का माल बिक रहा है, गाड़ी खाली हो रही है, तो ट्रेडर क्यों मेहनत करेगा? ट्रेडर क्यों नीचे फार्मर को एजुकेट करेगा? इफ यू वांट टू एजुकेट फार्मर, द वन थिंग इज गिव मनी.
We will do that.
One more thing, education
Yes.
is needed,
Yeah,
but
WNO.
that is all related with money.
Yes, sir. WNO will look into that. He will do that.
I would like to say something. Since the inception of WNO, we are planning to create neem fruits collectors society like on Amul pattern, as Mr. Abhishek is saying, depending on their moisture content, this, that, we have to fix the price. Dr. Rekha, we've always wanted someone to come with us, and we will create the societies, you train them, because we don't have much of manpower. We have all minds with us, but we need manpower and the experts who can train them. We made documentary. We will be sharing the QR code so you can see the documentary, and based on that and your experience, we would like to enter into some collaboration to increase the efficiency of fruit collection, quality fruit collection. That's what we are planning since years. Thanks to you that we will do something about
Yeah,
it.
definitely.
Thank
Thank
you.
you. Thank you very much. It was a wonderful panel discussion. It was a really very thought-provoking. Many of the points will be incorporated, and then we will revise our V2 version. Simultaneously, we will take up the input from the pharmaceutical sector and also cosmeceutical sector for inclusion. And on behalf of WNO, I would like to thank Mr. Jaipuria, Mr. Ravindranathan, Mr. Vikayat, and also Mr. Gopal Swami for helping us preparing the version 1 of this status paper. Thank you very much, sir. We sincerely thank you for all your inputs. And now I request Mr. Mark to make the...
That this is just a beginning. We have collected the information. Now what WNO will do, we will analyze the situation and identify the steps that need to be taken for further action. At the NGO level, at WNO level, and at the administrators, government of India and state government levels. Once we identify these steps, then we will get back to them and we will talk about the further action.
Yes, we are looking forward to new members. Thank you, ladies and gentlemen, for being so active in this participation. As discussed, we have made a beautiful documentary to address the issues that are currently in the NEEM industry and how to cope with them. We have experienced people who gave us interviews, and that documentary is right now available on WNO's YouTube channel. You can scan the QR codes. Unfortunately, we have a connectivity issue here, so we won't be able to play the documentary. But on the right side, you can see the QR code for our English documentary, and on the left side, we have the QR code for our Hindi documentary. ये documentary हमने 2 भाषाओं में इसलिए बनाई है क्योंकि नॉर्थ और साउथ की प्रैक्टिसेस में हमको बहुत अंतर नज़र आया. बहुत सारे सजेशंस आए जो कि स्पेशली उन प्रैक्टिशनर्स के लिए है, उन ट्रेडर्स के लिए है जो नॉर्थ में फ्रूट कलेक्शन करते हैं, प्रोसेसिंग करते हैं. साउथ की जो प्रैक्टिसेस हैं वो नॉर्थ से काफ़ी अलग हैं. इसलिए सभी की कन्वीनियंस के लिए इसको हमने 2 भाषाओं में बनाया है. Please watch these documentaries and give your valuable feedback. These QR codes will be displayed over here during the tea time, during the lunch time. Please take full advantage of it and please give your valuable feedback. Now I would request everyone that you can enjoy your tea, you can enjoy the trade fair, and we will meet back here after some time. So what time exactly? After 10 minutes, we will be gathered back here again for the technical issue. 20? 12:15, okay. 12:15 we will be back here.